Oh gosh,
All right.
I've got to just change this around.
Yeah, change it into.
Oh, and I've got to swap screens as well. So, oh, damn it. Where does that happen? And then I do.
We can see it. We can
Yeah,
see
no,
it.
but I don't want to see the other one. Hide presenter view. Great. All right, everybody. So this is lecture 2, I'm guessing, for this year, which is to look at soil monitoring and the physical and chemical assessment of soils. So I'm just going to give you an overview. There is so much detail to soil. The more you go into it, and depending on what area of environmental science or agriculture you're into, there is so much in soils. I mean, soils are amazing that they are able to hold chemicals in place. They hold ions and they hold organic molecules in situ, and we use those for growing. Um, Also, that creates a problem with pollution. They can also hold in pollutants. And there's an awful lot of interest in soil at the moment, particularly since we've gone to carbon trading. They've introduced the carbon trading scheme that has to be done through the federal government. So you start with a piece of land which you, for years, well, for 200 years, nobody has paid much attention to Australian soils from the point of view of agriculture, and have allowed them to become depleted in carbon. So they weren't particularly high in the first place because Australian soils are very old. It's one of the oldest continents on Earth. So they are depleted, and we live in a dry climate, and most of our topsoil, every time you see a cloud of dust coming in from the west, that's the topsoil blowing off and off into the into the Tasman Sea, into somewhere between here and New Zealand. So we do lose an awful lot of soil. Now, the emphasis at the moment now is amongst agriculture is to try and retain that carbon and actually use it to gather CO2. If we can convert the CO2 from the atmosphere into organic carbon in fields, then we will increase the carbon content, we'll increase the productivity of the soil, and we will also save a lot of CO2 that we're currently letting off into the atmosphere. So it's a win-win situation. The problem is with monitoring it, you have to keep an eye on what people are doing. And from a point of view of somebody who might be interested in doing that, the issue you would have is that you want to find the worst. If you're going to start doing it, you need to start with the worst piece of land you can get your hands on, because your gains in terms of carbon testing and getting the carbon level up in the soil are really good over the first few years, and then they kind of level off. Like everything else, you get a diminishing return. So you're hoping lots of people who joined early are reaching that kind of plateau, and then waiting for the next technological advance which will allow them to leap forward and get more carbon into the soil. But they do level off. And if you've already been, what's annoying is if like our farm here on the campus, they've already been doing increasing the carbon in the soil just because it was so depleted, farm was dead, and they did it because it was the right thing to do. They've actually kind of stuffed themselves in terms of carbon trading and getting the carbon credits because the soil has already improved massively over the first 5 years. So when you start the scheme, you should start it with the most depleted soil you can get your hands on, if you're going to go into that. Anyway, let's have a look at some of this. So the um, the soil fits into this zone. Okay, it includes the root zone of the plants, and also it's down to the groundwater underneath the soil. And I will keep referring to the the campus because I also teach things into soils into agriculture, and that's what we're really interested in. Is we have a huge problem on the campus. If you've ever looked out there, the groundwater is extremely high because it sits on top of a clay layer that is either rock hard in when it's dry or like glue when it's wet. So um, but in both cases, it's impervious to water. So even if it's full of water, it's become like glue. So we're looking at this topsoil, this topsoil layer, which can be extremely shallow. What we have is an open macroporous substrate based on silicate, iron, aluminium, and silicate. Lots and lots of different forms of it depending on the pH and what rocks have decomposed to form that soil. So what you're looking at when you're looking at soil is rock which has decomposed into iron, aluminium, and silicate with a really interesting open structure with big pores in it where ions, metal ions, and other things can sit inside the chemical structure of the soil. But they do change, and it depends on what kind of rock has decomposed in order to form it. So the soil environment is this subset of this open macroporous this structure. It's a weathered geological material, and into it you put biological residues, which I've already talked about, but it can also be manure. All of the products that we put in there, the decomposing plant life and the decomposing animal life too, these biological residues then stick inside this open structure and they come in contact with the Earth atmosphere. One of the key things in here, when you're looking at soils, is that at the top you will have, it's likely to be well aerated, especially if it's got lots of roots in it. Lots of oxygen can get into it, so that creates oxidizing conditions for the iron. If you look at the soils of Australia, they're bright red. That's because the continent is so old and they've oxidized the iron from iron 2 to iron 3, and it's all gone completely oxidized. There's lots of oxygen around in the top layers of the soils, and there's not really a reducing zone. If you're looking at more waterlogged conditions, and I come from Europe, you'll see that the soils are darker, kind of bluey red color. Some of them are a deep dark brown color. That's because they contain mostly iron 2 minerals, and that's because less oxygen can get into the soil. So the color of the soil will change depending on how much oxygen and water gets into it. So it's all weathered materials. Okay, so soils are pretty amazing. The further you go into, the more you realize that how much amazing chemistry actually goes on there. So, they're key to water purification because the water has to filter through if it filters, lots of the metal ions will be taken out of the soil, out of the water by the filtration of water through it. So it's like a huge filter bed. I talked briefly about carbon sequestration, that if we can get organic carbon back into the soil, we can increase its productivity and we can also trap CO2 from the atmosphere. My background in archaeology, there's a lot of stuff on soil types, on how it affects preservation. If the soil is reducing, that means not much oxygen can get into the soil. And then you get the preservation of amazing things like leather, bits of wood get preserved as if they went in yesterday. Things don't rust. But the majority of oxidizing environments cause the disappearance of all organic materials from the soil. And so, yeah, depending on the soil conditions, you'll get different preservation. We get construction materials. These are the kind of uses we put in. I'll just leave you to have a look around that. But there's an awful lot of iron chemistry you can see in here, metal iron chemistry, metal ion accumulation, which causes some problems, of course, if you can get bioaccumulation of some heavy metals. One of the concerns that we've had is that when water is recycled, it tends to build up cadmium in it. Cadmium is not very soluble unless it's in the presence of organic molecules, and in certain conditions you can get quite a lot of organic molecules. If you get a lot of algal bloom, it picks up the cadmium goes into the water supply, it gets into conditions where it's not mobile, but then it can be mobilized by organic materials in the soil. And so this cow, which is maybe grazing very low levels of carbon across the surface, will bioaccumulate cadmium in it. If you think grazing animals will bioaccumulate these metals, which is a bit of a problem. Okay, and ultimately, of course, let's have a think about the CO2 cycle and the nitrate cycle. We've got the soil is what controls how much carbon is trapped in the soil, but also its release. Because when it decomposes the carbon dioxide and water, and you did get carbon dioxide release, and so we had global carbon concentrations in the atmosphere. One thing I'm not going to cover, but is really, really interesting if for people who are into it, is that the soil is the source of, plants are the soil are the source of most of our pharmaceuticals, but also in the soil itself, there is a huge range of microbiological fauna which are on the roots of plants, which enable them to trap and reduce carbon. Yes, reduce carbon. I'm just trying to think whether it's oxidizing or reducing. It's reducing. Reduce the carbon and trap it. So you take nitrogen from the soil, from the air, and turn it into nitrate. Now, lots of those nitrate reducing bacteria live as mycorrhizal fungi on the roots and the hairs of plants, like it's illustrated in here. And there's a huge amount of work going on at the moment to see if we could isolate particularly efficient mycorrhizal fungi and then inoculate the field with them to get carbon, to get better means of trapping nitrogen in the soil and therefore giving the plant nutrition rather than using inorganic nitrate and pouring it on. Because there's another aspect to soil, which is that many of these minerals that you see in here, like phosphorus particularly, we can put large amounts of phosphorus into the soil. In fact, in the old days, they would throw superphosphate around in order to get the plant to become, the field to become productive, without much thought of what happened to that phosphorus. And what happens to a lot of it, phosphorus is not particularly immobile unless it's in the it's there as ammonium phosphate or sodium phosphate. If it turns into calcium phosphate, it's almost totally zinc phosphate. It's almost totally insoluble. So you can trap phosphorus in the soil. It's doing no good in terms of getting plant growth. It's also contaminating the soil, and lots of Australian plants have evolved to be in very phosphorus poor soils because that's what Australian soils are like. They're extremely low in phosphorus, so the phosphate levels are very low. They've acclimatized to that or evolved to manage that, and if you give them too much phosphate, you end up poisoning them. So there's an awful lot of things in soil. I've covered about 4 different discipline fields almost in doing that quick summary, some of which I'm familiar with. Some, like the microbiology of soils, is a big research area of great interest, but I don't know much. It's not my area, but I'm really interested in it. Okay, so let's have a look at how soil forms. As I mentioned, it's from disintegrating rock. So weathering of rock occurs through wind and water hitting the surface, and no rock will be perfect. No no geological material is ever completely perfect in its formation. The closest you would get the granites. Those don't have many fissures in them, but anything like limestone or sandstone will when it's formed, it will have small, tiny micro cracks. And water is extremely good at finding its way in. Because that water is not neutral, it's not pH 0, pH 7, it will either be slightly acid or slightly basic. It will eat away slightly at the rock, and over time, over geological time, this rock will disintegrate. Now, once it's disintegrated to a certain point, then seeds that are blown onto the surface have enough broken down material to grip onto and bury themselves into and start to grow. Even at this stage, where we've got the organic material that's got mixed in with the soil in a very, very thin layer, this looks something like the no, if you, this is almost like the soil on the campus, except this big blobs here would be one great big clay bed. But get the idea that the large amounts of rock get broken up, the soil seeps in, organic material can seep in here, and the roots then can accelerate this process of breaking up the lumps. Ideally, you'll move to get the A and B horizons in here, where you'll get residual rock in this area, but this, what we're now going to call in the A horizon, we're going to call topsoil, would contain high organic content, and the transformation, the minerals in here will depend on the bedrock and the broken down rock of the sea horizon. It will depend on those still. You will get acid or basic soils depending on the breakdown, but they are not chemically the same as the bedrock. I don't think I've got a slide of what the crystal structure of a soil looks like, but it is a great big open structure, and what's amazing with it, it's got these channels, ion channels, which hold positively and negatively charged substances in them, and the identity of those depends on the acidity. Okay, so what does all that depend on? Well, it depends on the parent material, which is what I've just mentioned, but also the climate will determine how harsh the climate is, will determine the rate of weathering. Time, of course, in Australia being old, it's a long time. Topography will change because you'll get different amounts of runoff if the ground is not flat. Time is in here, and then you've got plants and animals. The animals, since we've had macrofauna, they have changed the soil composition, and basically through manure or through their usage of grazing, they have a huge impact on the underlying soil, both in terms of its organic content, but also its compactness. Soils in the world, yeah, there's a huge variety. This is a really, you can just see a few of them. So I'm just having a quick look at it. I haven't looked at it since last year, but you've got ferrosols, calcisols. They're all, depending on what they're based on, they're classified basically on the rock that caused their formation. Now, don't just think all soils form like I just showed you. Some of them are then transported alluvially. So a lot of the soil on the campus or on the river farm down towards the Hawkesbury, it's rock that's been transported a long way, and then it got caught in the river bend and it just settled out. So the soil that you'll get in a particular area does not necessarily relate to the local bedrock if it's been transported by alluvial transport, and that could have happened either since the last ice age or the previous ice age, or it could be due to glacial deposition. Okay, so here are some, and you can see the different types. These are the main classifications. So we've got young soils from volcanic deposits which are degraded, sandy soils, which are broken down sandstones, almost no soil development. They're just basically grains, and a lot of Australia, of course, has got that. There's any topsoil that could have formed has disappeared. Ferrosols, just strongly weathered. They're chemically poor, but they are physically stable as a subsoil. So if you could put organic content into them, you could get a reasonable soil out of them. This is like talking about alluvial deposits. Fluvisols come from redeposition of material formed one place and deposited somewhere else. I'm not going to go through the whole list, but there's what you will see they all have in common is that they are iron aluminium silicates. Okay, and then we've got the vertisols, these clays, and clays are very, very fine particles of soil that are broken down, that they then get a particular structure to them that allows them to take in huge amounts of water and huge amounts of other metal ions into them. They soak up and swell and shrink. Here's the arrangements of soils in Australia, which you can see. And then let's have a look at this. Let's have a look at the horizons. So our basic organic horizon O, the O layer. Then we've got our surface layer, which is still topsoil, but it's not, it's tending towards the subsoil level. It's what gets it to be called the A level is the amount of oxygen. It's basically the oxidized zone. When we get down to subsoil, you're looking at areas which are oxygen depleted. And then we go down into the substratum and the bedrock from which they're formed. And you can see that they can change quite a lot. Just having a look at those forms. These are all from China. Yeah, you can see how it forms in layers, and each of these horizons will be either a change in the how much oxygen and water is the penetration of oxygen and water into the soil, or it could be a relayering of an alluvial deposit on top. On every scale, this is really important, is that there's no such one thing that is soil. And I talked about them being iron aluminium silicates, but they're not just minerals. And in fact, the mineral deposition part of it, which may control the pH of the soil, is probably the least interesting part of it in terms of what you might do with the soil and how it might impact on an environmental. We might use it in environmental solutions to problems. So it will trap a lot of water. It will contain some organic materials depending on how good the soil is. May contain more. Having more organic material in there causes it to use up some of the oxygen. It becomes more reducing. It then, for many of the minerals, becomes more acidic. It will also then, the metal ions become more mobile. It's more useful as soil for agricultural purposes as a reducing soil than it is an oxidizing soil. So although the mineral content might be 45%, and air might be 25% of the space, the minerals are important, but they're really just to hold the rest of the stuff. If you just had minerals, you'd be looking at the beach like a sand. There's nothing, or a desert. What makes it a soil and makes it useful is the other things that get trapped and held within the soil. Okay, so why would you want to measure or assess it? Let's have a think. What would you want to do? What we want to do is characterize it. If we want to know what to grow, where to grow it, then knowing what kind of underlying soil we've got is really important. They'll show you how the soil evolved, but they'll also show you what the kind of, once you've classified it, if you've got substandard soil, how could you improve it? It'll give you an indicator of what kind of improvements can be made based on, once you've determined what kind of soil you've got. So the soil mapping in Australia has been quite extensive now, and this is I think this is the New South Wales version. There is a national one as well, a federal soil survey, and you go out and you report on what your soil profile looks like, and you put it into the Department of Industry and Environment, DPI. And they're producing soil maps which are extremely detailed for areas. And you can see that some parts of New South Wales, well, in fact, there's no part of New South Wales. The mountainous areas, of course, have got the biggest variation because they've got the basalt tops, the basaltic rocks of the tops of the mountain, plus they've got the underlying sandstone that has been eroded away. So you get quite a lot of variation because that was a volcanic. The formation of things like the Blue Mountains was the seabed at one point. It's a raised seabed that's then eroded. So the basalt layer would have formed over the surface, and then it's got eroded away into the valleys that we see. And it was on a set. So we had a sand, which became sandstone, a basalt layer on top. That got raised by some volcanic action, became a raised surface, and then that's eroded away to form the mountains that we have now. So that's why we get such a big variation in soil types, because there are two different types of soil in there. There are different types of underlying rock. And you can see Then if you look, particularly in the Blue Mountains, if you get onto the basaltic rock, you will see a different environmental profile in terms of plants growing than if you see on than you have on the sandstone. It's really marked that as you travel across the mountains, you can see the different soil types by the vegetation which it supports. So making a link between the soil, underlying soil, and the care of the environment is really crucial, because there's only certain plants. If we're looking at endangered species, we have to make sure that they grow in the right soil background. Okay, so what can we measure? Well, we can measure the soil horizons by looking at our profiles between the organic layer and then down to the bedrock. We can look at the color, which I've already mentioned, which tends to go from red down to bluish grey, especially when you get to clays. And that blueness, that trend from red to black and blue, comes from the oxidation state of the iron. So iron 3 is red, so there's lots of oxygen available, low moisture. You'll get red soils, just like we get red sands all over the country. If you go down into the ground, as the oxygen levels get lower and maybe the water levels increase, then you will get oxygen depleted areas, and the iron 3 minerals will turn into iron 2 minerals, and they will change from red to bluish grey to Prussian blue. That will also change the chemical composition, because different layers of soils, they can contain different amounts of hold different amounts of carbon in terms of organic content. The bluish grey ones contain more organics than the red ones, where the organic content is pretty much oxidized. That also changes the pH, and it will also change which metal ions are free and able to move within the soil. We can look at the color distributions by size and contrast. That will tell us what kind of mixture of minerals we've got and what size distribution we've got. So the texture and particle distribution comes into that. Identifying what the soil is will also determine its porosity and therefore its water holding capacity, because it's the voids in there that will voids in the soil which will hold water and retain it. And so if we wanted to know about things like evapotranspiration and what level of soil, what level of growth we can support on there, knowing the water density, or the porosity, is important in determining the water holding capacity. And then that takes us into the water drainage and infiltration. The more porous the soil is, the lower the levels of drainage, and that will impact on other things. If we're applying, or if somebody is applying inorganic nitrate as a fertilizer into a soil with very low holding capacity and very low porosity, that nitrate is pretty much going to go straight through the soil literally like a dose of salts, and into the river system. So this impacts in terms of the soil type and its characterization, impacts on its water and the water holding capacity, but also the holding capacity of the minerals that may have been applied as a fertilizer, and whether you're going to get problems of runoff. So these things can be measured in a laboratory. You can measure the particle size distribution, the types of sand, the various proportions of the minerals, how much water it will hold when it's saturated, and how much water will it hold when it's dry. What's the difference in mass, and what's therefore its porosity. We can measure the pH of saturated soils and its electrical conductivity. And electrical conductivity will tell us how many metal ions are present. It won't identify the metal ions, but it'll tell you if it's the level of salinity in the soil. When you get a water saturated paste and you work out you put the conductivity meter in, and it'll give you a good indication of the salinity of the soil. If we put them in soil into an oven and burn it, we can measure how much organic carbon and how much nitrogen. If we measure the gases that come off, we can therefore get a measure of the organic matter component of it, and the richness of the soil in terms of what life it will support. and then the carbon can, and we'll also get the carbon exchange capacity for doing things like carbon trading. So what should all that information tell you about? These are the things that we get: the thickness, organic carbon, particulate matter, exchange for ion exchange. We want, one of the things we want to happen is that we want to swap out one metal for another, but we have to be very careful because sodium salts, if we swap them out, then contribute to an increase in salinity of the soil, and you can end up with poisoning it with sodium chloride because you're exchanging out the other metal ions. So this is important for the long-term viability of the soil. It's pH, electrical conductivity. And you'll notice again, I'm skipping a bit on soil enzymes, which are incredibly important, but it's not my field. So one thing that's really important is that the soil must be able to buffer. So it filters out materials that are unwanted in the soil, but it buffers the pH so that we don't get wild fluctuations in acidity and basicity of the soil, of the environment, because the soil, with its various metal ions and its various amounts of organic compound, are capable, if it's a healthy soil, of filtering and buffering pollutants and maintaining a steady pH, which is really what we want. For growth, of course, we want nutrient cycling. So how do we go about doing it? Well, basically you go out with a soil auger and test in various areas. So a typical borehole, this one's a more serious one, will go down deeper. But you can see they're quite simple apparatus, will give you a soil profile. If you want to sample and work out water holding capacity, you can't do it with these. You'd have to use a bigger diameter sphere, like literally like a cake cutter, for cutting cakes or pastry. It's a circular thing that you take a plug of soil, which would be about 7 centimeters in, 5 to 7 centimeters in diameter. You take that away from the soil, you wrap it immediately in cling film, you measure it, you dry it in an oven, you measure its water capacity, how much water you lose when you dry it, you saturate it, and then you do it again, and you can get the water holding capacity of the soil. So here we are. There's the borers that I was talking about. These are wider borers. The problem with this kind of borer, there's the ones for doing the soil moisture. The ones that like this, they churn up the soil as they go down, so it's no good. You do need to get a representative sample of the soil. Soil varies across a field, across an individual paddock. This paddock may look flat, but there will be different in the subsoil. There will be undulations of the bedrock, which means that the soil will vary not only in its composition, because it will flow, the subsoil will move, but also in its oxidation state. The amount of oxidized material in there, the amount of air that can get in there will change, because the water will be in the low areas, the waterlogged areas will be in the wetter areas of the soil. So you do need to get a representative sample. Now, in order to do that, you need to plan out grids and then work out a random pattern. Once you've started doing some sampling, and if you're getting consistent values from across a whole paddock, then you can say, okay, that's fine. We don't need to do any more sampling, but you may have to return to do more sampling if you find out that you're getting large degrees of variation from your samples from the same field. Here's the kind of analysis. Starts with sieving, and this is the pH testing kit that you do in the field. These are to remove plugs of soils so that we can measure the water capacity. And one thing that's incredibly important is a good labeling system right from the start. It's so easily overlooked, but you have to be organized, because you've just got bags of soil coming back to the lab. You've got no idea intrinsically where they came from. Make sure you've got where in the field they come from well marked out. Now that's a lot easier now. We've got GPS, so you mark where you are in the field, but also the depth, because soil will change with depth. So you need to get the depth profile written down, and so good documentation, because it is so easy to lose track of what the soil samples are when they get back to a lab. When you're talking the difference between gravel, which we all know about what gravel is, and we all know what clay looks like, but they are actually the same thing. They're just the insoluble component of the soil at different sizes. And that's why we're sieving, wet and dry sieving, in order to work out what's the size distribution. Because the gravel will be highly porous to water, and the clay, like we have underneath the campus here, is almost water impervious. You can't get water, it just traps the water, which is why we get so many flooding problems with flooding. And you can see how you classify the different types of soil, and they're basically on their size profile. So a sandy soil have large pores. It will drain really well. They have high amounts of drainage, and a clay, by contrast, will be much smaller pores. So we'll have a greater porosity in the clay soil. We'll have less porous because of the big lumps. These are less porous, and this will hold more water. So the clay can have an enormous water holding capacity. It may not always be useful, like it's not useful here, and it can block drainage like crazy. So if we have a sandy soil, we'll look at, you put your sandy soil type in, you'll get very thin layer of silt at the top. And the different types of clay will have different components. So a clay soil will contain, about between 50, anywhere between 50 and 100% of clay. That's very fine particles. So the pore space is what determines the water hold capacity. If you've reached saturation or past saturation, the water will then drain down through the soil. If you're at the field capacity, it means it's got the maximum amount of soil it can hold without losing any. And then if you've lost that, the water, the interspatial water, you've then got no water capacity in the soil, and you'll get the death of the plants that are in there. There's nothing being held. Now, do remember that the clays, so the soils, are structures which hold large amounts of water, especially if they're clays. The clays can have their own drawbacks in terms of growing stuff, but in that they're so dense, you don't get any drainage through them, but they will hold an awful lot of water. So porosity, the amount of gaps within the soil, the density, like how much does it weigh per cubic meter, how much does the water infiltrate, and what's the amount of water, total amount of water holding capacity, are all things which are going into an Australian soil survey, and which we regularly measure to determine the health of the farm. Now, soil porter for pollution assessment. There are lots and lots of things which can damage soils in terms of making them less productive, or making them toxic, or just because they're big open silicate structures. The very thing that makes them useful for holding water means that it holds everything else, and so you can get accumulation of toxins in soil. So it may be that somebody is only polluting with a small amount of toxin going into the soil, but because that is then getting trapped within the soil structure, it's accumulating and can reach massive layers. So let's have a think about that. In surface impoundments, like in pits and ponds and lagoons, we can get deoxygenation happening. So if you've got a pit that doesn't drain, or the levels drop to a certain level, it starts to go stagnant. You'll see that the oxygen capacity, or the oxygenation of that water, reduces, and at that point a whole load of other things become soluble, which are mostly harmful. So you can get I'm just trying to think of an example of one, but things like, well, there's a whole heap of minerals that become that are highly toxic, but they become particularly mobile once you've got them into deoxygenated environments. So if the oxygen content of the soil of the water in the pits or ponds or lagoon goes down, then suddenly you get increased the mobility of these ions, which are then highly toxic. Many pesticides are not easily broken down. Let's not think too much about nerve gases, but chlorinated solvents are very regularly used in bleach and stuff. And these things accumulate. They bioaccumulate in soils because they get trapped within the open structures. So the kind of pollutants that we would get, landfills where you're chucking toxic substances into a small concentrated area, of course, that's a big problem. Landfill sites where there's no monitoring, all sorts of rubbish gets thrown into those, including heavy metals. If you think too much about the area where I live next to Olympic Park, that's been a toxic waste dump for more than for at least 100 years, and there are some very, very nasty substances. There are notices up on all the bays around then telling you not to fish, not to eat anything from the water, from the sea outside, and that's because everything was being dumped in there. So heavy metals are a big problem because they may bioaccumulate in the soil. Radionuclides, they've been thrown into Homebush Bay. They've dumped everything in there. So this is just some of the flows. And if we just go back to the accumulation of things like salinity, that can accumulate because you're adding sodium phosphates as fertilizer. The phosphate then becomes insoluble, say, as iron phosphate in the soil. Now the sodium is loose and it can add to salinity. So you can get a salinification of the soil, not because of what was put on, but because of what it turned into when the more insoluble component dissolved out. If you want to sample the pore water, you put in a lysimeter like this into the soil, and what it does, and you suck, basically pump it back out, and the water will go through the boundary. You hope you get the soil water out, and then you suck. Basically, you're sucking it out. There's a vacuum above here. It will pull the water in. Now, this is a really good way of getting soil because it doesn't of soil water because it doesn't. You're not doing anything chemically different to it. You're just pulling it out, pulling the water, literally sucking it through, like through a straw. Why do you want to measure the soil moisture? Well, the soil moisture is about 35% of healthy soils. The mineral component's about 50, and the rest of it's air. The soil moisture content determines the soil water holding capacity, and the more we can increase the soil water holding capacity, the more healthy the soil is. So the more we find that it's correlated to the organic compounds that we can get in. The more organic compound we can get trapped in the soil, the more the water holding capacity is, the less susceptible it is to evaporation, especially on a continent like Australia, where we've got long periods of drought. So our advantages in doing soil moisture is that we can work out how successful it's an indirect measurement of how much soil organic material there is in there. So we measure that by volumetric water content. It's the volume of water divided by the total volume of the sample. So the volume of the water we can measure by drying it. So we take a soil sample, we dry it in an oven, we weigh it, dry it in an oven, the water is lost. The water capacity then can be worked out from the volume of the sample compared with what you've got before it was wet. We can measure in situ using sensors. These are really good, but they have to be calibrated. Don't just think you can go out into the soil, just leave them there and forever. You do have to keep track of that they are giving you sensible answers, but basically they're an electrode, and what they're doing is they're measuring the conductivity, because all groundwaters contain soluble ions. and it will measure how much how much electricity is passed between these two electrodes. And the wet soil will have a higher current between them and therefore a lower voltage. So the voltage will drop. The drier the soil is, the less transmittance of electricity there will be between them, and you'll get a higher output voltage. I don't know what the neutron method is. I think that's neutron activation analysis. I'm going to leave that one out. It's not common, and you'd need a specialist piece of equipment in order to do it. But electromagnetic sensors, of which these are mostly mostly what we're looking at. Yeah, these are the instruments that you can use. Oh, here's the neutron probes. Okay, so this is how you're doing. Okay, this is called neutron activation analysis. And what you're doing is you're putting, if you're doing the thing that makes it handy is that it is highly accurate and highly precise. It's based on the propensity of water to slow down fast energy neutrons. So if you have a radioactive source, which you lower into the soil, and then you measure the radioactivity, the changes in radioactivity with depth, that is due to the water molecules slowing it down. So you can count the number of neutrons, the neutron decaying counts, because these are radioactive isotopes. You can count the number of decays along the tube, and it will tell you how much water content there was in the soil. Now, the beauty of that is that it's non-destructive. You can go back and you can go measure in the same place. The problem with many moisture content, things like taking this off to a lab, measuring cores, is that you get one off and you destroy your sample when you do it. So this is good for in situ measurements. The problem is you need a radioactive source. That needs a special licenses. It's not something you can just buy off the shelf and every farmer will have one. They're much more likely to have soil moisture sensors that are like this, which are basically a pair of electrodes. Why do we want to measure the soil salinity? Well, when we water plants, they transpire, water evaporates, and that means that even if low level salinity was present in the soil with which we're doing the irrigation, because we've got constant evaporation, it means that the salts are accumulating behind. If we're not getting sufficient rainfall in order to wash those salts down into the soil, or the soil is compact and not very good at draining, you will get accumulation of salts, and you'll get problems of salinity and poisoning of the land due to the accumulation of irrigation water. The way you measure it is using a conductivity meter like this. You can measure salt concentrations pretty well using a conductivity meter. Now, why measure the soil pH? The mobility of different ions depends on the pH. Okay, so this one is showing you how mobile nitrogen is as nitrate. You will see that it's most mobile at high pHs. Phosphorus, likewise, is more mobile in the region of 6 to 8, the pH 6 to 8. Calcium and magnesium, the mobility of calcium drops off with low pH. It's an optimum around 6 and 7. If we want to make nutrients available to plants, they have to be in a mobile form. And so the region 6 to 7 you can see is pretty much optimum for many things, but not for others. So the micronutrients like iron, manganese, zinc, cobalt, and copper, these are very poorly mobile at high pHs and can get trapped. And so don't think that just because you take a soil sample, you dissolve it in a very strong acid in order to get it into solution, and you measure the ion content of that soil, that gives you a measure of what's happening in the actual soil. It will tell you the total mineral content, but not at different pHs. different elements will be have different mobility. So as you can see from these viola graphs, the mobility around 6, 7, 8 of most things is the same, but it's not for the trace elements. And so you can end up with a soil that's trace element deficient because you're in a region, say 7 to 8, which is neutral pH. A typical soil pH might lie here. That means that iron, manganese, zinc, these transition metals are not very soluble, and if they're essential to plant growth, then there's very low concentration of them available. What you do is you take your sample and you mush it up, put it into an ultrasonic bath to get it and measure the pH. It takes a lot of measurements. It seems simple, but a lot of because the soil is such a complex mixture of organic material and inorganic material, the mobility of the soil will depend on not only of how much is in the base soil, but what's the organic content. And so, for instance, I've done a lot of measurements in my time on phosphorus. It is incredibly difficult. It seems simple. There's a reasonably good method for measuring phosphorus in a laboratory, but the results can be absolute nonsense because phosphorus can be present not only just as phosphorus P3 minus, but as a whole range of other oxidation states in different forms. And some of them get highly locked in and very difficult to remove. And so one of the most interesting things you want to know is phosphorus concentrations for the fertility of soils and their condition. Phosphorus levels go up. I was particularly, when I was doing archaeological work, interested in the phosphorus content because phosphorus is a good indicator of human activity. Phosphate levels go up around human activity, so we were doing phosphate surveys. It's incredibly difficult to do though, because if there was a lot of organic material, you get a completely different oxidation state of phosphorus than in the form of phosphate. So you got readings that depended not only on the human activity in there, but the soil profile as well, and how oxidizing the environment was that we were looking at. So these are not simple. Manganese things. Manganese is a difficult element to measure because it can be present in so many different oxidation states. Iron is not simple either. The more simple ones are zinc, copper, and cobalt. And sulfur too can be present in different oxidation states. So these things are deceptively simple in how it looks like. They require huge amounts of samples. You have to do repeat measurements. Okay, now this oil organic matter is everything that's not inorganic in there. That's the silicates and the aluminiums and the metal ions. And that's the carbon, hydrogen, nitrogen, phosphorus, and other things, which if you look on the periodic table are up in the top right-hand corner. And they're the basic forms of nutrients that the plant requires. They contribute to the resilience of the soil, and it's something that we are actively involved in, is trying to increase the carbon, nitrogen, phosphorus, or not phosphorus, but the carbon content of soils is part of carbon sequestration and a response to climate change. So the biological functions depend on carbon, nitrogen, phosphate, the organic content of the soil. The physical functions, that's the inorganic material that's present in the soil, which basically contributes to its structure. And as I've mentioned before, it influences how water is held. Some of the clay minerals can hold huge amounts of water for a very small amount of stuff. So that also, and this also alters its thermal properties. So inside the soil, we've got physical functions which come from the organic materials, from the inorganic material from which it's derived. We've got chemical functions in that the structure of the soil can change its holding capacity. Water holding capacity influences how the pH is controlled, whether it fluctuates a lot, because metal ions can be held and released and buffer the pH to a certain level. And then the biological functions, how healthy the soil is, depends on the carbon content. Now, if we want to measure carbon content, We can do this in what's called a LECO machine. It's a CHN analyzer. I have been known to call this, and got in trouble in the past for calling them random number generators. It's deceptively simple. You take a soil sample, you sieve it, you put it into a small sample inside the machine, and you burn it. And as you burn it, you have columns which absorb carbon dioxide, nitrogen dioxide, or and water, and the water holding and water. So you can absorb those things which are given off when you burn it. This is basically an oven, and it burns off the samples. However, the variability of the soil is enormous, and you can see how this person is sieving the soil before they put it through, because you've got to get it to a homogeneous sample. Otherwise, if you just take random samples from here, you're going to get completely variable results in your analysis. And as I said, I've got in trouble for calling these random number generators, because getting consistent results is not simply a case of take the soil out, sieve it, and shove it in here. You have to be very careful on what you're putting in. But in principle, you can measure the organic content of the soil. We can measure that using remote sensing, but they're pretty sophisticated bits of kit, as you can see here. And this is one's measuring near infrared reflectance spectroscopy to try and get the soil sensing data. You can measure the electrical conductivity. So you've got the electrical conductivity will tell you how much salt, the salinity of the soil. So you can use remote sensing, but you also need to go and ground truth it, because the machines do go out of calibration. This is a conductivity meter that has been you walk across the field. I can't remember what make that is, but should know. You walk across the field in a pattern, and what you're measuring, it's got two coils in here, and one coil is sending out a signal. It's bouncing into the soil and bouncing back up here, depending on how many conductive ions there are in the soil. The signal from one end to the receiver to the from the emitter to the detector will get slowed, and that slowing down will give you a measure of how much salinity there is in the soil. They're really good, very simple to make. You get lots of exercise walking back and forward, but the readout is really simple to do. You can take direct measurements. These direct measurements into the soil will have a soil probe and will give you metal ion concentrations. They'll have ion specific electrodes on here and tell you how much of each of the what trace elements are present in the soil. More and more we're using remote sensing for these purposes. It's a rapidly growing area, and it's being driven by the fact that now we have computers that can capture huge amounts of data sets and process them really quickly. So there's a whole load of things. The capture technology, we can use satellites and drones. The sensor technology has improved massively. Different forms of spectroscopy, that hasn't changed so much. But what really has been is the processing technology and the GIS, which puts these things into layered maps and enables you to produce an image of a subsurface feature produced from a remote sensing tool, and process that into a sensible. Now that used to take ages. I mean, it was what was held everything back. But now with robotics, we can get these drones to fly patterns across the fields and collect the data very quickly. What we can get is the subsurface variation, and you can see the different types of radiation that you might want to use depending on what question you want to ask. So if you're looking at subsoil texture, you can use gamma rays, electromagnetic induction, resistivity, visible near infrared. There's ground penetrating radar, which will go down deep into the soil. So these are the different techniques you get for the different types of data. Now there is a huge interpretive step in here. You don't get just a number saying, or a flashing light saying, blah, 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 this means this. It does take interpretation. And the skill, which hopefully some of you will go on to get, is in interpreting the signals from these things. They're not simple to do, but they're really interesting. And we can use, one of the main ones I've been looking at is looking at the use of ground penetrating radar. What's improved this massively is that it can be done remotely using GPS as a remote sensing system to tell you exactly where you are. So they do have harvesters that work off remote systems. You do still have to have a driver in them in case the thing went haywire as a manual override system. But basically the person can sit there and working off the GPS satellite system because we've got now two-way communication systems. It used to be that only that the farmer would only be able to tell exactly where their position is, but now we can retransmit data back from the mobile station and keep updating it. What we get is a map that looks like this, and this one will give us an indicator of yield. I'm just trying to look. Oh, this one's measuring. So what's happening as the harvester goes through, it's measuring data, the collection, the mass of data that's collected in the harvester, and then mapping it, relating it to where that was collected in the field. And if we did that through looking at the field underlying the fertilizer mass balances, this is the protein levels of the grain it's producing, you can see that this area of the soil is producing the highest yield, but it's got the lowest protein content, so it's poor quality. So you can relate all sorts of things across the thing, because they can all be measured at the harvester level. We can measure moisture, we can have sensors for things like oil content, and we can then relate a measurable component that we're interested in to the position in the field where it was collected. One thing you do get is this anomaly. You see here, this kind of line of dots, this pattern of dots. That's a product of the fact that they were of the harvester going across in that direction, right to left, and then turning around and giving us those dots. You can get artifacts which are not real, but they're artifacts of the data collection system. If you measured that with the harvester going vertically instead of horizontally, which you wouldn't do because you want long runs, but if you wanted to do it that way, then these things would disappear. You'll see them here as well. So we can get a true reading of fertilizer demand based on that. Electrical conductivity, I mentioned that you can use that conductivity meter here. It measures the moisture content. It also measures soil type and salinity. That can be related to cation exchange capacity, of how much capacity is in the soil to exchange one cation for another. We can do radioactive isotope measurements. Igneous rocks all contain radioactive isotopes, and we can detect those gamma rays in up to 0.7 meters into the soil. So we can run get the soil underlying geological soil type by running a sample across the surface and measuring the radioactivity. Okay, let's have a quick look at the new technologies that are available and what's changing this. For looking at vegetation, the big changes that are coming are 3- and 4-dimensional LiDAR. That's putting a radar up into a drone, and it fires off light. So it's light. It's like radar. It's not radio waves, but it's light that's being fired, and it will give you the canopy height. So you can measure vegetation growth by flying a drone across the surface and then collecting the data. So we can scan plants. Is that a link? See where that goes. Well, perhaps shouldn't have done that. Let's see where it's going to take me. Whoa. Yeah, I think we'll leave that one. Okay, so that will take you to an advert for the plant phenomics. Okay. What have I gone? I hope I haven't got rid of my presentation. Oh, no, here we are. So we can scan the fields. This is an area, and what's driven it is the increase in computer capacity. All of these things have been driven for the last 20 years. The processing speed and the amount of data that a computer can hold have increased so rapidly that a whole lot of things have become available. And you can imagine that you want to do 3 and 4, you want to do growth patterns of vegetation in the field. You can do a 3 and 4 a 3D LiDAR survey will tell you the height. Doing it in 4 dimensions with multiple images over multiple days will give you a time scan. That's a huge amount of data. Just the data holding capacity and data processing is what held this stuff back for years. But now it's pretty much the sky's the limit in what we can do. So it is relatively easy now to capture high resolution imageries. The clever bit and the bit that you're interested in as the scientist is what do they mean and how are we to go about using them. Anybody can collect the data, interpreting it is the clever bit. So what we can do is work out organic content, we can do chemical analysis, we can do all of that by remote sensing as well. So we can measure the organic content of soils, either by going out and doing soil samples, which takes a long time and requires a laboratory, or we can do it by measurement. Okay, I'm going to have a quick break there if that's okay, guys, just for 5 minutes. And the reason I want to do that is I'm just going to go and nip into the chemistry lab and just make sure everything's okay for the chemistry lab. Are you all right with that, Ricky, just for 5 minutes? Oh, it's gone to sleep. Okay, well, I'm going to do it anyway, and I'll come back in a moment. Just give me 5 minutes. I just need to check on the lab. Thanks. Going to skim through this a bit quicker than the last part of the lecture. Okay. So we talked about the soil functions, and we're interested. What we're really interested in is soil organic content. If one of the problems we face in this country is that we live on one of the driest and oldest continents on Earth, and most of the organic material has either through weather conditions or just the age just blown off and is somewhere underneath the Pacific. So what's happened, if you get what we left with is the soil mineral particles, but the soil minerals on themselves don't contain much life, which is why Australia, for a large part of its landmass, is not particularly fertile. What we need to get is stable humus. We need to increase the soil carbon or soil organic content between 1 and 6% of the total mass. And that's what our aims are in agriculture at the moment, is to try and increase soil carbon. If we do that, we will also increase soil microbiological biomass. And then we get active fungi and we'll start to get more fertile soils. And that's the aim of the last 10, 15 years and where we're heading. We're trying to increase the soil carbon content. The soil organic and the soil organic material aggregates inside. It's not a homogeneous substance. Soil is not a homogeneous substance, and it will contain macrobiological samples and mycorrhizal fungi living on the outside of the fungal, so outside of the roots. So we get aggregates. We don't get. It's not a homogeneous substance, and the closer you look, the more non-homogeneous it becomes. So the complexity of soil comes from the basic. I have to be careful with the word basic. Both the fundamental components, like things like quartz, which is silicon dioxide, but that it is able to trap organic material. Water, clay particles can hold huge amounts of water and huge amounts of air. So inside, it's not a homogeneous substance, it's a heterogeneous substance. Depending on the soil type and its location and its depth, they will more or less homogeneous. And if you're looking at fine particles, very, very fine particles like clays, well, they can become pretty much homogeneous. But the majority of soils are heterogeneous. Okay, so if we're looking at the scale of where the organic components are, what we're really interested in terms of research work is the mycorrhizal fungi that we're getting here, and we're talking about the stage of microns. So within a millimeter of soil, we have a huge amounts of variations of both in size particles, but also of the mycorrhizal fungi that you will get active inside the soil. Now, the soil biology happens within the soil matrix, within the organic compound, and I will admit that my area is in the inorganic compounds, but the research effort in soil is now highly focused on the organic components, which go down from earthworms right down to viruses within the soil, and particularly the fungi. So 1%, the living 1%, the soil food web supports all land-based animals and plants. Basically, without soil, we're stuffed, okay, on every single level. Either right from insects down to bacteria and subparticles. The food web is dependent on its carbon. And one of the particular problems we have in Australia is that we are on one of the oldest continents of the world, oldest and driest, and a lot of this carbon content has become oxidized and disappeared. So, of the species that are found on Earth, 25% of them are all found in Earth, inside the soils. That's things like fungi, bacteria, plants, protozoa, algae, hundreds and thousands of species of nematodes, all found in the soils. But without them, without that, anything goes wrong with this, there's no chance of supporting any macrovertebrates or any other forms of life on the terrestrial surface. So our life on Earth depends on the health of soils, is basically what that's saying. If you look at the areas of biodiversity across the world, you can see that they fall into the equatorial regions are extremely fertile, tropical regions. They get less fertile as you go out. Australia being humongously big, has very fertile areas up in the north and the east coast, but then huge areas of non-fertile, of very little biodiversity in the soils. So this is the biodiversity present in the soil. Because we know that the soils of Europe are pretty fertile, but the amount of biodiversity as you move away from the equator goes down. How do we assess that? Well, we take a soil sample and we'll need to take it into the lab. You need to hand sort everything to get rid of things that you're not interested in. you can then centrifuge it, you wet extract, dry extract, and then you can cultivate the particular. If you're looking at the mycorrhizal fungi, you can cultivate and then do your molecular and biochemical analysis. Molecular approaches are becoming increasingly important, especially since we recognize the importance of mycorrhizal fungi as an expression of biodiversity and also of genetic potential. So, and as computing power again has gone up in processing genomics, the power of, or the possibilities for doing microbiological work and DNA and RNA work on soils has increased. So that enables us to work out the diversity of what's present in the soil using DNA amplification. We still rely on GCMS, which is what that is, in order to do analysis of the other compounds in there, like the presence of phospholipids. This is a GC flame detection system that's measuring the profile of bacteria and fungi and mycorrhizal in the soil. Soil sample. It's not a simple process, and you need a lot of samples. We can also we do the gross separation of soil fungi. Those are good indicators of soil health. And then we work out the soil web and its function, which is incredibly complicated. Okay, because you get different orders of consumers of decaying materials. If we want to measure simple measures of the soil functions and the nutrients available, there are plenty of very quick small samples. If we want to do decomposition study and see how the soil is able to decompose, how quickly how it decomposes materials, we can do that by bagging. And I've done this with bodies, actually looking at decaying bodies. That's part of forensic science. We can also get these strips, and these will turn different colors depending on the presence of, say, ammonium present in the soil as things decompose, or as the presence of sulfate. So it will absorb. This ones will absorb nitrate. These will absorb cations, like those, and these will absorb the positively charged anions. So anions and cations have got the wrong way around. So they absorb positive and negatively charged ions, which you can then take away and take back to the lab for measurement. If you want to monitor microfaunal activity, well, we can. This isn't my specialization, so I'm not great on it, but we can actually trap the plants and trap them within a sensor and take them out. What is more my field is measuring the CO2 flux coming from the soil, and the respiration, things like ammonia. Really like remote sensing of soils, because you can keep going back and doing your measurements. You can take as many measurements as you like, whereas taking soil samples back to the lab changes them in many ways. It's very difficult to retain them intact and get them back to the lab, and the things that you do for testing of one type, you can't, then destroys its potential for being used in another type. So you end up with hundreds and hundreds and hundreds of samples. So cost effectiveness becomes really important when you're doing soil analysis. There are a myriad of things that you can be measuring, and millions of ways of doing it. So these are ecological indicators produced. It was a very well-known paper, actually, basically scoring the samples on how resilient the soil was. Sampling becomes really important. So a sampling plot will have to have a randomized pattern of whatever you're collecting. So if you're, say, collecting earthworms, you're going to collect it from 3 different places. You can't measure that because you're digging up the earthworms. You can't do that when you're measuring the water infiltration. So that has to be done in a separate spot. If we're looking at the soil example, soil sample, we want to look at the soil composition. We will need an area that's got to be decided on for that. If we're looking at microarthropods, we have to measure those separate again. So all of our soil samples and a representative number of soil samples must be collected from our grid, but they have to be collected from different places from the others. So you can see that, and this isn't even counting the number of analyses that you're going to do on these for microarthropods and for the soil cores, and it just becomes enormous. So the number of samples from a simple 8x8 grid would have generated huge numbers of samples. It's immensely complicated. If you want to determine where to sample, that's really important. You need a representative area. You need to look at the macrofloral indicators of health or lack of health, and then if you're trying to, you have to ask a research question which is directed. You can't just go in there and just go into a site and just say, I'm going to measure everything, because it will cost you a fortune. You're not guaranteed not to get any answers at it. You've got to go in with a research question. So you're looking at ecological indicators of threats and then looking for evidence of those through the data that you collect. Just blind testing is not going to work. There are millions of different analyses. You will not get any answers out of it. So choosing your area is vitally important. If we don't have soil, we end up like the moon. So, or the deserts, these arid desert areas. Okay, so it's really, really important. I'm going to finish there, Ricky. That's 1 hour and a half. Are you still there?
Yeah, yep.
Okay, so I'm going to finish there. If there are any questions from anybody about soil sampling and testing, if they haven't gone to sleep. Okay.
You'll have a chance next week. So
Yep.
Richard will be in the lab,
Okay. Yep.
computer labs next week, and I see Anna's here. So Anna and myself will take the lecture next week, face to face. So thanks everybody. I'll package this up and put it up on Views this afternoon.
Okay.
Thanks, Richard.
All right. No worries.